Hello everyone, I am Aarti Dwivedi and welcome to this detailed lecture on controlled release drug delivery systems or CRDDS, an innovative and evolving field in pharmaceutics. Controlled drug delivery systems CDDS, an innovative approach in drug delivery presented by Aarti Dwivedi, course B.Pharm 7th semester, subject NDDS. Let's begin by understanding the fundamental shift from traditional to controlled drug delivery. Introduction. Traditional drug delivery methods often result in fluctuating plasma drug concentrations, leading to suboptimal therapeutic effects and increased side effects. Let us begin by understanding the problem that controlled drug delivery systems aim to solve. Traditional drug delivery methods, such as conventional oral tablets or injections, often result in fluctuating plasma drug concentrations. This means that after administration, the drug concentration in the bloodstream rises rapidly to a peak, then falls as the body metabolizes and eliminates the drug. These fluctuations are visible in the graph shown here. Notice the peaks and troughs. What is the problem with this? When drug levels are too high, above the therapeutic window, there is a risk of toxicity or side effects. When drug levels fall too low, below the minimum effective concentration, the drug may no longer produce a therapeutic effect. This is called subtherapeutic dosing. Therefore, this pattern leads to ineffective treatment, increased chances of side effects, and often the need for frequent dosing to keep drug levels stable. This not only affects therapeutic outcomes but also burdens the patient with strict dosing schedules. This is the central challenge CDDS is designed to address. Controlled drug delivery systems are designed to deliver drugs at a predetermined rate for a specified period of time to achieve a consistent therapeutic effect. In contrast to traditional systems, controlled drug delivery systems are designed to release the drug at a predetermined rate and over a specified period of time. This ensures that the drug concentration in plasma remains stable within the therapeutic window for longer duration. Take a look at the graph here. It shows a controlled and consistent drug concentration over time, represented as a flat line, which is ideal in clinical scenarios. There is no sudden peak, no dangerous drop. Such a controlled drug release profile offers several benefits. First, reduces the frequency of dosing. Second, minimizes the risk of side effects. Third, prevents underdosing. This is the core concept behind CDDS: to maintain therapeutic drug levels consistently and effectively, thereby improving clinical outcomes. CDDS represent a significant advancement in pharmaceutical technology aimed at improving drug efficacy, safety, and patient compliance. Controlled drug delivery systems are not a modification of existing drug delivery, they represent a major technological advancement in pharmaceutical sciences. They are designed not just for convenience but to optimize the therapeutic index, that is, to maximize efficacy while minimizing toxicity. CDDA offers increased efficacy, enhanced safety, and improved patient compliance. Increase efficacy by maintaining optimal drug levels. The pharmacological effect is sustained over time without fluctuations. Enhance safety. Controlled release avoids the peaks that could lead to toxicity. Improved patient compliance with reduced dosing frequency and more predictable outcomes. Patients are more likely to adhere to their treatment plan, especially in the treatment of chronic conditions like hypertension, diabetes, and psychiatric disorders. CDDS plays a crucial role in long-term therapy management. Let's now analyze this very important graph that illustrates the difference between immediate release and controlled release drug delivery systems in terms of their impact on plasma drug concentration over time. This graph has two curves. The yellow curve represents an immediate release formulation. The orange curve represents a controlled release formulation. Now let's first look at the immediate release profile. As you can see, the drug concentration shoots up very quickly, reaching a sharp peak shortly after administration. This is because the drug is released all at once and is rapidly absorbed into the bloodstream. However, this also leads to a high peak plasma level, which may enter the toxic range, especially in sensitive patients, followed by a rapid decline as the drug is metabolized and eliminated after some time from the body. The drug concentration quickly drops below the minimum effective concentration, meaning the therapeutic effect is lost unless another dose is taken. This kind of release leads to fluctuations in drug levels, which can cause side effects at high concentrations and therapeutic failure at low levels. Now, let's turn to the controlled release profile. Notice that the plasma concentration rises slowly and reaches a moderate peak. More importantly, it maintains the drug level within the therapeutic window for a much longer duration. This controlled release system avoids high peaks, thereby minimizing toxicity. Sustains plasma concentration over an extended period, thus maintaining therapeutic effect. Reduces the need for frequent dosing, which greatly improves patient compliance. Instead of dosing every few hours, a controlled release formulation might require only one daily or twice daily administration. This is particularly valuable in managing chronic diseases. Now, Let us examine this graph which compares multiple conventional doses with an ideal controlled release profile. On the y-axis, we have plasma drug concentration, and on the x-axis, we have time in hours. The graph displays two types of profiles. Blue solid lines for conventional immediate release doses, orange dashed line for ideal controlled release profile. In a conventional dosing, the patient takes multiple doses at regular intervals. Here, dose is administered around hours 0, 12, 24 and 36. Each dose causes a sharp spike in plasma concentration right after administration, reaching a peak, followed by a rapid decline as the drug is eliminated. Between doses, the drug level can fall below the minimum effective concentration, resulting in subtherapeutic periods. These repeated peaks and troughs have several drawbacks. Fluctuating drug levels can lead to side effects during high peaks and treatment failure during troughs. Frequent dosing increases the risk of missed doses, which further disrupts therapy. Poor patient compliance, especially for long-term treatments. Now, look at the orange dashed line, which represent an ideal controlled release system. Gradual rise in drug concentration after administration maintains a stable, steady level within the therapeutic window for an extended duration and avoids the toxic peaks and subtherapeutic troughs seen in conventional dosing. This means reduced frequency of dosing, more predictable and consistent therapeutic effect, improved safety profile, and better adherence by patients because dosing is simpler. Let's now study a fundamental concept in pharmacokinetics, the plasma drug concentration versus time curve. This curve explains how drug levels change in the body after a single dose and introduces important parameters such as Cmax, Tmax, area under the curve, and the therapeutic range. This curve has three major phases: absorption phase, post-absorption phase, and elimination phase. First, absorption phase starts immediately after administration. The drug concentration in plasma rises as the drug is absorbed into systemic circulation. The slope of this curve depends on absorption rate. The faster the absorption, the steeper the curve. This phase ends when the drug releases its peak plasma concentration known as Cmax. Now, post-absorption phase. At Cmax, the absorption rate equals the elimination rate. This is the turning point of the curve. After this point, elimination begins to dominate over absorption, causing plasma concentration to start decreasing. Now, elimination phase. Here, the elimination rate exceeds absorption rate. Drug levels steadily decline due to metabolism and excretion. Now, let's define the important parameters shown here. First, Cmax. Cmax is the maximum plasma concentration achieved by the drug after administration. Clinically, it helps assess the potential for both therapeutic effects and toxicity. Second, Tmax. Tmax is the time taken to reach Cmax. This indicates how quickly the drug starts producing its maximum effect. Now, area under the curve. It represents the total drug exposure over time. Larger area under the curve means more overall exposure, which relates directly to bioavailability. Now, onset of action. The time from administration to when plasma concentration first reaches the minimum effective concentration, the lowest level that produces a therapeutic effect. Duration of action. Duration of action is the time interval between when plasma concentration first crosses minimum effective concentration and when it falls back below minimum effective concentration. Before we explore controlled drug delivery systems, we need to understand a fundamental term: drug delivery system. A formulation or device that introduces a drug into body and enhances its efficacy and safety. This definition gives three key aspects. First, formulation or device. Drug delivery system could be a tablet, capsule, injection, transdermal patch, inhaler, implant, or even a nanotechnology based carrier. Second, introduction into the body. The method by which the drug is administered means oral, parenteral, transdermal, pulmonary. And third, enhance efficacy and safety. The goal is not just delivery, but delivery that improves therapeutic effect and reduces harm. Now we come to a key pharmacokinetic term, bioavailability. It is defined as the proportion of the administered drug that reaches the systemic circulation in its active form and is available for a therapeutic effect. This tells us how much of the drug actually makes it into the bloodstream after administration and is ready to act on its target. Now, factor affecting bioavailability. Several factors influence bioavailability. First, route of administration means IV or oral. Second, drug properties like solubility, stability, molecular size or polarity. And third, physiological factors means gastric emptying rate, intestinal motility, pH of the GI tract, presence of food or other drugs. Now, next is controlled release. Controlled release refers to a drug delivery system that releases the drug at a predetermined regulated rate over an extended period of time. In the graph, controlled release generally produces a smooth curve compared to conventional release, but its exact shape depends on the design of the formulation. Next, sustained release. Sustained release is a subtype of controlled release where the drug release is slowed down and extended over time. but not necessarily at a constant rate. In the graph, sustained release is represented by a curve that starts higher than zero order and then tapers off, showing a gradual decline in release rate. Next, zero order release. Zero order release describes a drug release profile where the same amount of drug is released per unit time, regardless of how much drug remains in the formulation. In the graph, zero order release appears as a flat horizontal line after initial absorption, indicating constant plasma levels. Now, comparing the three profiles using the graph. First, conventional release means pink curve represents sharp rise to peak followed by rapid decline, large fluctuations in plasma levels. Second, sustained release means orange curve represents extended duration of effect but gradual decline in plasma concentration. And third, zero order control release means yellow line represents constant plasma level throughout the dosing period, most desirable but more difficult to achieve. Now, let's refine our understanding of control release and sustained release by adding a real-world factor into the picture, the drug elimination. This graph shows us how both release types behave when the body's natural drug clearance processes are taken into account. First, controlled release. Controlled release represented by the blue dashed line in the graph. Zero order means the drug is released at a constant rate for a defined period, regardless of the drug's remaining concentration in the dosage form. Now, sustained release. Sustained release represented by the green solid line. First order release means the rate of drug release is proportional to the drug remaining in the dosage form. So, release slows over time. Both controlled release and sustained release eventually show a decline because once the input rate decreases or stops, drug elimination dominates. The differences lies in how stable the therapeutic level is before elimination takes over. Means controlled release, flat stable profile before decline. Sustained release, rises and falling curve, more fluctuation. Let's take a closer look at zero order drug release. The top graph shows a perfect straight line. This means the same amount of drug is released per unit time. For example, 5 mg every hour. It doesn't matter how much drug is still inside the dosage form, the release rate is constant. The bottom graph shows a horizontal line. This means the drug's plasma concentration is perfectly steady over the dosing interval. Achieving this stability happens when drug input rate equals drug elimination rate.